important role as well in the life science. Okay. So with this little thing, let me go to the, because some students are there. So I will start with a very brief, slides which are undergraduate slides, something. Carbon is a basic element of light, and all of the students, they know carbon has, two allotropes like diamond and graphite. But what happens, diamond, all of you know what is this, and graphite, all of you know this structure, this layered structure. Diamond is a very hard thing. Structure is like this, but you see, then we have, well, this is the structure of diamond in details. I'm not going into detail. Then graphite, this kind of layered structure, all of you know, and the separation is kind of 0.142 nanometer. Distance, the lattice separation and the distance between the planes is about 335 nanometer, 0.335 nanometer. Next, we have to see these two, not only the diamond and graphite, but there is one allotrope called the fullerenes. Fullerenes is made of C60 or C70 or C74, and their structure is kind of buckyball structure. I'm coming to that. That is, these are the people who got the Nobel Prize, in physics, Rice University, for their discovery of this particular material called the buckyball or fullerenes. And let's see, this is a structure of fullerenes, C70 or C60. That's why it's called the buckyball. You see that all carbon atoms, but they form kind of buckyball structure. They could be nanotube also. I'm coming to that later on. I'm not going to this. Then, if I want to say more about carbon things, then we have to know something about nanoscience and nanotechnology. And all you know what is nanomaterial, 10 to the power minus 9. And some of these devices have demonstrated application in everywhere, like medicine, electronics, robotics, and energy.
সঞ্জীব দা আপনার স্ক্রিনে দেখুন নিচে একটা ওই বার সব সময় থাকছে স্টপ শেয়ারিং এর পাশে হাইড করে দেন। হ্যাঁ, থ্যাংক ইউ।
ঠিক আছে?
হ্যাঁ হ্যাঁ,
এখন ঠিক আছে না?
হ্যাঁ ঠিক আছে, পারফেক্ট।
হ্যাঁ স্যার, ঠিক আছে, গুড। So these nanomaterials, those who are students can get an idea of what is the size about nano. If you use a grain of sand that is 1 millimeter, then you if the lower and lower, if the red blood cells have 10 micrometer, or human hair that is about 150 micrometer, and then if you go lower and lower, and transistor to 35 nanometer, and then protein, usual the globular protein or elongated size protein. They have something like this 10 nanometer diameter or 10 nanometer in length if you consider the whole structure. Then you go to the carbon nanotube, little less, 5-10 nanometer. The nanotube, I'm coming to that, the diameter. DNA, the diameter is 1 nanometer. An atom, all of you know, 0.1 nanometer. So these are the scale, 1 nanometer, 5-10 nanometer, the scale of nanoscience or nanomaterials or nanotechnology. So get an idea of this, and then let's say why it is so important. This is for again an undergraduate slide, that if you have a 1 centimeter, this area is 6 square, and then you divide it into a cube having say 1 nanometer side. Then if you calculate how many cubes will be there, and if you calculate the area, you will find that 1 gram maybe area will be a area of a football ground, a cricket ground. So much surface area, and that's why these materials are very much useful for catalysis. All you know what is catalysis, to enhance the rate of the reaction, and you need lots of surface area for this. And this is one important area of nanomaterials that used as a catalysis. Okay. So apart from this, they're subdivided into nanocrystals, nanopowders, and nanotubes. And as I go forward, these classical nanomaterials, sometimes called the 0-dimensional, this is nanoparticle, and then 1-dimensional nanotubes, nanorods, and nanowires, 2-dimensional called nanofilms, nanolayers, nanocoatings, and 3-dimensionals, the dispersions of nanoparticles, bundles of nanowires, and things like that. And I'm giving examples. This is carbon nanotube. So this is a allotropes of carbon with a cylindrical structure and can be capped on the ends with the buckyballs open-ended, composed entirely of sp2 carbon. This is carbon nanotubes, and there are various. These are properties that are diameter 3 to 9 nanometer, lengths, efficient electrical conductors, can act as both thermal conductors and thermal insulators as well. And this is the toxicity. Something is there toxic, and then these are called single wall nanotubes. Single wall nanotubes, there's one wall, but we can have double wall, like multi-wall nanotubes are also possible. So people have prepared this kind of nanotubes, single wall or multi-wall, and they have lots of application in various fields, starting from chemistry, physics, material science, life science, everywhere. And these are various methods to prepare nanotubes, but I'm not going into details of that. One of the important methods is laser ablation or chemical vapor deposition, ball milling, and art discharge. I'm not going into that. Applications are very, very useful in our daily life. Electrical, energy storage, lithium batteries, hydrogen storage, biological, they can act as sensors. They are used for AFM tips or DNA sequencing also. All these materials are used. So huge applications. I'm giving importance to biological application sensing. Many spherical nanoparticles have been fabricated for bio application. Nanotubes offer some advantage. Relative nanoparticles, and these are all very useful in biological sensors. Then I come to the very important discovery in physics, and Nobel Prize goes to scientist and Andrew Joim, and this is 2010, and they the first thing showed that this is graphene called, that is one layer of graphite. If you think of graphite layer structure, they prepare one just layer, and carbon-carbon bond length is like this. Graphene sheet stacks to form graphite with an interplanar, already mentioned. That if you just take away one sheet, that is called graphene, and that was first done by these people, and two-dimensional material graphene, and called the Nobel Prize. And these different allotropes of carbon, therefore, are related to graphene, in fact. So if I call this is graphene, one layer, then graphite will be stacked graphene, as you see there, or nanotube will be rolled graphene, and fullerene will be wrapped graphene. So this is one way of understanding what are the allotropes of graphene in terms of graphene. Graphene is stacked, the graphite. If you roll it, the nanotube, and then if you wrap it, it's called the fullerene. So that's all about graphene things. I'm not going into details of that. There are lots of applications, as you see here on the screen, that everywhere, graphene quantum dots, including graphene biodevices, inert coating, the transparent terminating electrode, solar cells. It's very important in solar cells. Electrochemical devices, single molecule gas detection, many applications of graphene are there. That is why they got the Nobel Prize. Orpon, am I audible clearly?
Yes, sir.
Okay, and screens are visible, right?
Yes, fully visible, sir.
Okay. Then I go to the something which is very much useful in our daily life. That is called the quantum dots. First, quantum dot, nothing but your family with semiconductor, but this is semiconductor nanocrystals. So I told of nanocrystals, nanoparticles, nanocrystals, semiconductor. They are made of many of the same materials, ordinary semiconductors, mainly combination of transition metals and other metalloids. And unlike ordinary semiconductors, the quantum dots are extremely small, on the order of a few nanometers, and very nearly zero-dimensional. And I may go through a little bit of this. The semiconductor energy level, all of you know what is band gap and carbon conduction band, as well as valence band, and energy band levels, semiconductors. But these are some things, and the light is emitted when electron is in a quantum dot decays from an excited state to the ground state. You can get the light, and electrostatically delineated quantum dots also can be prepared, and they have lots of applications. Optical storage, light emitting diodes, particularly so important in preparing light emitting diodes, optical storage, and these two things are so useful in the advancement of in spectroscopy and other material sciences. Organic dyes, quantum computing, that's coming a very big way, and these materials, these quantum dots are used in security purpose as well, and solar power. So these are the very few important aspects or usefulness of this particular quantum dots. And next, from this one nanoparticle, I go to the something else, because so talking about nanomaterials. So let's see, and people probably know what are the. Any students are there? Can this identify these two people? Any students are participating?
Yes, sir. Many students are there.
Okay. Can anyone tell me the name of these two persons? Give the figure. Okay, I don't know. This is DNA structure and done by Watson and Crick. And Crick was a physicist, Watson was a physiologist, and both of them awarded a Nobel Prize. And when they discovered this Watson was only 24 years of age, and Crick was 37 years of age. And another lady actually done the crystal structure, but they did all the calculation and manipulation to find out the DNA structure, actually double helical structure, and that is the birth of molecular biology. That is looking biology at the molecular level. Not only these two persons, the another person who was so famous for this is Linus Pauling. You have heard the name. He got the Nobel Prize in chemistry as well as in peace, but he also first person to identify what is called the sickle cell anemia, and the mutation you're talking about these days, coronaviruses, all times you're talking about mutation. First mutation was discovered actually by Linus Pauling, and he is also considered the father of molecular biology. But these two people mainly, the structure of DNA, and after the structure of DNA, we come out, lots of things come out, and people have started looking at the biology, as I mentioned, at the molecular level. And next important discovery came out in the ribosome structure. In the meantime, lots of structure of proteins have been discovered by crystallography. Ribosome structure is the Nobel Prize again, was won in 2009, and we are proud that one of the Indians got that. Venkatraman Ramakrishna is fully worked in crystallography group, the physicist from physical chemist, and these two persons also, Jonath, the lady, and the Thomas citizen. So they all were awarded Nobel Prize for this particular discovery, ribosome. It is so important. I am not going details of this. These are the functions of ribosomes. If you are interested, we can tell you the later. It is a very important discovery. Now after this, we go to the another discovery, and I call them the magic molecules, and I include two molecules here. One is green fluorescent protein called GFP, the other is ferritin. Now I want to say ferritin, green fluorescent protein is isolated from a jellyfish, that is a jellyfish. I will show you the picture. And ferritin is in our body, which stored iron. And these days, you probably are aware of the fact that if someone has a COVID, then the ferritin has been tested in the blood. How much ferritin you have in the blood, it's a very good marker whether you are cured or not, because that controls the blood coagulation and things like that. So ferritin is I'm coming to that. It's iron storage protein in our body. But what is green fluorescent protein? These three persons again won the Nobel Prize for this discovery of green fluorescent protein from jellyfish. So why it's called green fluorescence? Because this protein, if you shine light, ultraviolet light, then you can see a green emission. Normally, protein, all of you know, contains various amino acids, and out of them, tryptophan or tyrosines, they are the our phenylalanine. These the aromatic amino acids, and mainly they emit from tryptophan residues, and the emission comes around in the ultraviolet region, like 340, 350 nanometer. But this particular protein won the Nobel Prize because they emit in the visible region, and a huge application in this later on the molecular biology, medicine, cell biology as a biological marker. And you can see this is comprised of 238 amino acids and obtained from jellyfish, and they emit blue light or green light. Two types are there. So this is, I'm not going to details of this, how this bioluminescence occurs in that. This is one of this. I think if you shine light, then you get this. And then obviously you could see that structure kind of this is the why is it people are interested, because they got the Nobel Prize. Why the green fluorescence? Now because of this factor, I'm coming to that. They have a structure of this. This is the actual structure of a can shape. Reporter gene advantage, non-toxic. This is a protein, so non-toxic, intrinsically fluorescent. So lots of application in fluorescence microscopy, genetic engineering, and as I mentioned, is a useful in the biomarker. But why is this? This is a very useful figure, that absorption and then emission takes place. This is the green region. And why in nature you see this particular protein contains a serine residue, tyrosine, and glycine. One after another, the 65, 66, 67. Those are the locations of the amino 3 acids, and they form somehow a compound like this chromophore, and this chromophore, when you shine light, emits in the blue or green region. That's why it's the green fluorescence protein. But look at this. This occurs in nature. So amazing thing, and that was produced in jellyfish. So people who was isolated, got the Nobel Prize because this has a heavy useful applications in everywhere, particularly in in medical science, as I mentioned, and in genetic engineering, microscopy, fluorescence microscopy, this reporter gene. So very important discovery of green fluorescence protein. If you just Google it, you will find lots of publication papers out of this, and there are so many useful in our daily life. And then this is a figure again, emission from some insects like firefly. You're familiar with the firefly light, that's called the bioluminescence. Firefly, you can see the light of firefly in the dark. They don't need any excitation from the outside. They can emit light from their own because there is a metabolism, and they produce the some chemicals which is in the excited state, not in the ground state, and they come down to the ground state and emit light, and that occurs spontaneously within the firefly or within this insect. So this is called something called bioluminescence. Bioluminescence means this is the form of a chemiluminescence. Chemiluminescence, you don't need the excitation. Chemiluminescence means the product is formed in the excited state and comes down to the ground state. You get the light, and this is form of a chemiluminescence. Therefore, when light is released by a chemical reaction in the form of visible rays, fireflies, anglefish, and all the things. This I am mentioning this because this bioluminescence is used these days again in a molecular biology, microbiology, and many other. Particularly so important is known as the BRET. We are familiar with the FRET, which is resonance energy transfer, fluorescence energy transfer, but there is one subject. A lot of people are working on this. This is called the BRET, bioluminescence resonance energy transfer, where you can get some ideas or very important ideas in our body or in some cases some diseases, because this is non-toxic, totally non, and you don't have to shine light on this, and this has a very important future, and a lot of people are working on this. Now after this I come to the something that our laboratory is doing little bit, not details, that since I'm talking about the proteins, proteins, as I mentioned, it's contain tryptophan, but there is many proteins which contains more than one tryptophan, and then it is difficult to know the structure and functional aspects of this tryptophan protein, multi-tryptophan protein. And usually what people do, this is completely spectroscopy. They either steady state or time resolve, then the fluorescence correlation spectroscopy, or time resolve everything. But the problem in this, all these very is somewhat expensive things, but in the broadband and fluorescence, and always you get the multi-exponential decay, so difficult to address. What is the contribution of individual tryptophan to emission? What is the environment of individual tryptophan residue? So that is very difficult, and therefore what people do, you have to mutate protein. That means you have to remove one tryptophan by another residue, either tyrosine, and then you get the details of the different environment of the tryptophan, if it is multi-tryptophan. But problem is, if you get a mutation, then whole thing may change. As we are hearing these days, that one mutation can cause so many varieties of COVID vaccines. Similarly, things happen in the protein as well. If you mutate, your whole structure may change, activity may change. So how to do that, keeping this all tryptophan intact without mutation, and that's what my students or my lab is doing. It's called the triplet spectroscopy. If you take the protein in a very low temperature glassy matrix, then you can get the phosphorescence, and you can get lots of information without doing any mutation. So fluorescence spectroscopy as well as this gives us a lot of idea. And these days, you have heard of, I'm coming to that, cryo-electron. So if you freeze the protein at low temperature in some glassy matrix, protein remains same, and you can get a lot of information. So I just briefly mentioned what we are doing, and then also this is another area of research that people are doing these days, very important research, imaging technique and drug delivery system in aqueous medium, and obviously at physiological pH, because if you want to use as imaging technique for some for some disease, that means in a human body, or then you have to have physiological pH and must be in aqueous medium. Many imaging techniques that people have prepared is not in aqueous medium, solid state, but that are not very useful. And if you want to have aqueous medium physiological pH, then system that we have to use must be protein based, so that is non-toxic. Also, you can use nanoscience confined environment. For example, cyclodextrine, things like that. And natural protein cage. I'm coming to that. There are some called cage protein. There are certain proteins which has a cage inside, cavity inside, and that cavity has a dimension of nanocavity. That is called natural supramolecular system. I will come into that in details. And what you do, or what people are doing, they use the antenna effect. What is the antenna effect? Antenna effect is the enhanced emission of lanthanide ion, particularly in the 3 state, valence 3 state, and all systems are biocompatible because people are doing in this kind of platforms. And I will show you something. Before that, lanthanides are very useful features, attractive features rather. They give this emission in a very narrow band and away from the excitation light, large scopes as I mentioned, and very sensitive to chemical environment. Therefore, they are so useful. And many advantages of this lanthanide emission. Lots of people are doing this: advanced optical materials, telecommunication, LED, solar energy conversion, biomedical science, imaging technique, and this is what I call the antenna effect. Because if you excite a lanthanide, for example, you see the picture. This is the lanthanide excited and ground state. If you excite, you don't get the emission. But if it's connected to some ligand which can absorb energy in the ultraviolet or in the visible region, and that can transfer this energy from their triplet state to the, as I shown here, this is our antenna. That means a particular ligand. And if you excite this ligand, it goes to the singlet, goes to the triplet, and then they transfer the energy to the lanthanide excited state and get the emission. That's how you get the emission from the emission from the lanthanides. And since you are getting from this particular ligand, it is called the antenna. So whole effect is known as antenna effect because they are not exciting directly, and exchange takes place through Dexter exchange mechanism. So this is very useful. I am showing just one slide. How protein platform can be used. For example, drug tetracycline. If you bind to the 3 proteins like HSA or bovine serum albumins or alkaline phosphatase, you see that only in case of bovine serum albumins HSA, this emission is very, very high, whereas in alkaline phosphatase, the emission is very, very low. The same thing, and you can do the kinds of spectroscopy to find out the rotational correlation time, how they rotate within this, and you can see that. And if you do the bioinformatics as well, then you find where is the location, and see that in this case of BS or HSA, they go inside the protein, but in case of alkaline phosphatase, they bound to the surface. That's why the emission is so weak, because it is exposed to water, and water, what it does, which vibration they couple with the lanthanide and they degrade the emission. That's why it's not possible. But if you use these 2 proteins, they go inside, you can see the picture, and then you can analyze. So this kind of spectroscopy involves everything: lanthanide, antenna effect, then the protein triplet state, the protein singlet state, as well as the bioinformatics and all kinds of time result spectroscopy to find the rotational correlation time, and then measurement lifetime in D2O, the titrated water, so that you can get the hydration number and things like that. So this kind of research is going on to find the imaging technique in our body, and this can be used as a best platform for drug delivery, because this is protein, so non-toxic. So you can have various other drugs you can encapsulate within the protein and then use as a drug delivery system. I go to the next. As I mentioned, there are supramolecular system. It's called supramolecular system in nature, and sometimes they are called also nanoreactor in nature. And what is this? They are natural protein cage, nanostructure for drug delivery. You can see that assembled from subunits into spherical oligomeric structure. And this, when they assembled, the 3 distinct surfaces are there: exterior, interior, and interior nanocavity. And protein cage, they can encapsulate, protect, and deliver drug. And protein-based platforms, because of this, they are biocompatible, biodegradable, and very low toxicity. So they are available in nature. I'm showing you one diagram that I talked about, ferritin. This is the structure of ferritin. You see that? This is the monomer, and 24 of such monomer, they form a kind of spherical things. And you see that iron atom is like there. And one particular molecule, ferritin molecule, can store 4,500 iron atoms. and that is the source of iron in our body. And if you are iron deficient, then this iron must be depleted. So you have to have taken iron from the outside. And so that's why ferritin is tested in the blood these days for COVID patients, so that they don't get the coagulation, because that causes a lot of problems. So this is the ferritin monomer, and this ferritin, if you take out these iron atoms, easy to take out, and then you'll find a cavity, and that cavity can be acted as a nanoreactor, can be acted as a drug vehicle, drug delivery. So these are other examples of cage protein. Lots of cage proteins are there, and their dimension of this type, and you can see lots of materials can be incorporated for imaging purposes, photodynamic therapy, as well as MRI, as well as drug delivery used. I'm not going into details of this. Coming to this, only one example I'll give you, ferritin, and this is called apo. Apoferritin means when you take out metals, then the protein is called the apoferritin protein. See that cavity, and this cavity has 8 nanometer, 12 nanometer size. So you can have some material. You can, if you introduce some materials here, the nano size materials can be prepared. That's why it's called a nanoreactor, and can be used as a drug delivery. I'm coming to that. How do you do that? This is a way how we can open this apoferritin. It's just very simple. You just lower the pH. Suppose pH is at 2.66. There is a channel there, and then you put your metal, whatever the drug, or then it come back to the return to the normal pH. Then they could be encapsulated, something like this. This is light scattering experiments done on that, and you can encapsulate this, and then this can be used as a drug delivery. So take the protein, encapsulate, and then deliver. After coming to this pH, this is the system. We have done it to the U3TC system. That means tetracycline with europium. Europium is taken because you can see where it goes, whether inside or outside, by looking at the europium emission. So this is the kind of emission you can see, and that tells us that it goes inside. Okay, I'm not going into details of this. This is something, again, of lanthanides. Since I'm talking about lanthanides, these lanthanide materials, which is called DF hybrid complexes, that is the transition metal, as well as the lanthanides. They are called DF, and these are very important in material science, and single molecule magnetic magnets, or I'll come to that. People are using this, and we have done with the collaboration certain this kind of material. You can see the lanthanum, and this MM means they are either, that is transition metal and lanthanum metal, and they have a bridging ligand, other ligands. So this is a very important and interesting subject of DF hybrid complex. A lot of people are working on this. I'm showing you some of the emission that it gives us. For example, terbium or emission. These are some metals of this, and how they act as two ligands. They can transfer energy to terbium, europium, or neodymium. Neodymium has an advantage that emit in the in the near IR region, not in the visible region, and so you can have lots of things. Like imaging technique, you can develop imaging material. You can develop using these DF hybrid complexes. Also used as nano materials. They use as nano composites. These materials you can use. These are some of the, again, some pictures. I'm not going into details of this. So different DF system. Zinc terbium or zinc europium. So then next I come to the very small molecule now, from the big molecule to the small molecule. And this is so important these days, nitric oxide. And again, you see, these three people got the Nobel Prize for doing some work on the nitric oxide itself. And why nitric is so important these days? Because I think most of the participants have got their second dose, or at least one dose of vaccine, but people are trying to get nasal vaccine. Probably you've heard of nasal vaccines. Is you take like a nasal drop, you take a nasal vaccine, and people are working very hard to have a nasal vaccine, so that we can take this once in 2 months or 3 months and get out of this COVID situation. And this nasal vaccine contains this molecule, nitric oxide. The nitric oxide has properties like this. It contains blood flow, and platelets and memory. All this, there are lots of benefits and properties. So people are using this nitric oxide to develop nasal vaccine. That's why I'm taking this particular slides. And then something about come back to the bioinformatics, the proteins. You can do lots of bioinformatics to know whether drug is binding. A lot of people are doing this in the COVID situation. They have the structure of virus and they're taking thousands of molecules of drugs and trying to find out from bioinformatics where is the binding sites. And you can also calculate the binding mechanism as well as binding constant, all from bioinformatics. You can do that, using some. So this is that one drug that we call colchicine. How does it bind in the big protein? Two halves are there, so we can find this kind of thing. This, let me talk something about as Dr. Arpan Manna told me that say something about the modern things, and this is not a modern thing. A lot of people are using the stent, but again here, see the stent means the chemist people are required. Chemistry is required there. Biocompatible material, a tube where bare metal first, simple metal, and then we use the cobalt chromium alloy. And all of people know this used in coronary heart disease, artery disease, renal artery stenosis. So many use this, but this must be biocompatible. But this is some alloy, and these days some advanced polymer being developed to find the stent. So chemistry is everywhere. That's why I want to say, the students are there, I want to tell them that chemistry is everywhere. When I passed the higher secondary examination, I was thinking that I should take physics or mathematics because I got very high marks, about 100% marks in physics and mathematics. I got less marks in chemistry, but my mathematics teacher told me, go to chemistry, because if you do chemistry, then you have to do physics as well as mathematics enough. Lots of mathematics and physics are there. So go to chemistry and it will be very useful. So that's why I took the chemistry. And you see that even in stent, without the chemist, you can't generate this materials like stent. I'm coming not only stent, I'm coming to the batteries that we use in our body, battery used in your phone, everything is chemistry. And I'm coming to that. This is not only lithium batteries, other metals are dual ion batteries are used these days. Even you have to design batteries so that it can be used in our body. That's called the biocompatible batteries that have been developed so that we can have certain materials, certain chips in our body, in our brain. So those batteries are biocompatible. So important. Now, next I want to go through certain things which are very important in different fields. One of these is called MOF. Now, what is MOF? It is metal organic framework. Metal organic, this slides, the first few lines may not be readable. I don't know. These organic ligands as than some metals, and people are doing this. Lots of people are doing this. MOFs is called, in short, is known MOF. And this is a spongy material. There are cavity. There are inside, so that inorganic, organic will make them more flexible for some industrial applications in comparison to other porous materials such as zeolites. So these are porous, some spongy materials, very, very useful in many applications. I have draw kind of attention. The separation, storage, all this is very important, as well as catalysis. So I'll give you certain examples where MOFs are used. This is a slide. You can see that I'm going to the details of this. This is one. This is carbon dioxide capture. Very important these days. You have to remove carbon dioxide, and MOF can remove carbon dioxide. This kind of material can be prepared. I'm not going details, but they can encapsulate carbon dioxide in the material within that. And then methane storage, you can do that. Not only carbon, methane can be storage, and the beautiful molecule you can prepare. Some metals are there, and these organic parts, and you can have cavity, and you can have methane within this, methane storage. Then next one is, if you have a mixture, acetylene and ethylene separation, that can be done by some kind of MOF, as I mentioned and shown. You see here, this is a very interesting part of the synthesis. I can synthesize this kind of material. I'm not going to detail. You can see these are materials, zinc, something used, and you can get this. And finally, this kind of material can be used for separation. So interesting that you can separate acetylene and ethylene using this material, because they have a different kind of cages or inside cavities which can incorporate these gases, and then it's possible to separate this. Then there is another way, this is called the catalysis also. It plays very important role in heterogeneous catalysis. I'm showing one just example how this zinc MF or cadmium MF can be realized. And very many, in the catalysis they use, you can get a very high yield of compound using this MOF. And the participants must be aware of these today's that the chemistry Nobel Prize in this year have been given to two scientists who have done useful work or very seminal work in catalysis actually, and those are called asymmetric catalysis. The asymmetric things is very important in they have done few years back, and now people are using that for catalysis, and also people are using this MOF for catalysis. And I just want to mention that this asymmetric molecule is so important these days. If you have allergy problem, used to have drugs like cetrizine it is called, but these days if you go to the doctor, they will give the levocetrizine. That means the L variety of cetrizine, because that is so useful for useful drug in the sense of they can bind particular portion and can be act as a drug better way than the mixture of L and D. Similarly, these two people who got the Nobel Prize, they find out that one particular variety can give you the lots of better catalysts, and a lot of work has been done after that, and that's why the Nobel Prize has gone to them. And this is again MOF can be used as a catalyst. Apart from that, these are very much luminescent materials. If you have, as I mentioned, the DF hybrid complex, if you have metal like lanthanides and transition metal and form the MOF, then they can emit light in the green depending on the material, lanthanides, terbium or europium, or maybe neodymium in the near IR region. So they are very good luminescent materials useful for LED or other preparation. Again, may be used for the MOF as an imaging technique, probably provided they are biocompatible. Next, I want to touch upon something which is so important in these days. People are doing for the last 2 decades, the single molecule spectroscopy and microscopy. Single molecule, we all do the spectroscopy in a bulk solution, crystal or solution. There are lots of molecules are there, but what's single molecule? Can I locate the single molecule? If we do that, then it's called a single molecule spectroscopy or the spectroscopy or microscopy. Again, we have shown the idea of the site, chicken egg, then large protozoa, the chloroplast, mitochondria, proteins, viruses. These are sites, and then amino acids, and then atoms. So you can have a size in the back in the scale. These are those of 10 nanometer, 1 nanometer size, and atom is of the 0.1 nanometer, as I mentioned earlier. Also, so these are various kind of spectroscopy used, as you see: microscopy, scanning electron microscopy, transmission, then atomic force microscopy, compound line microscopy, for different purposes of different size molecules. So, and then human body made of cells and molecules of atoms and cells. Not only single molecule but single cell spectroscopy or single cell microscopy is very important. That is whether we can see a particular cell, not in an ensemble or just this particular cell, then we can understand very nicely or in a better manner that how the cell functions. And if you know the function of a cell, then you can find out many reasons behind many diseases like cancers and all this. So people are doing single cell spectroscopy or single cell microscopy, try to find out or isolate one single cell and try to find out its function and its environment, and then we can use this for various purposes, particularly the disease location, as the Francis Crick, and this is an important Francis Crick actually mentioned. If you want to understand function, study structure. Without doing the structure, you cannot do anything. But structure sometimes as a bulk material is fine, but if you can see something in a single molecule, a single cell, that will be very much important these days. And this, I'm coming to that. The trio of this scientist, they shared the Nobel Prize in 2017. What they did, this microscopy is called cryo-electron microscopy. If you've heard of electron microscopy, but this is cryo. As I mentioned, in our lab, I do the phosphorescence in a cryogenic condition. That means 77 Kelvin or helium temperature 4.2 degree Kelvin. And these people are doing the electron microscopy in the cryogenic condition. That means freeze the sample so that their structure is not destroyed at low temperature, and then do the electron microscopy. Now, what is the advantage? Advantage is you don't have to prepare the crystals, because crystal preparation is so important, so difficult. Preparing crystal of a protein, for example, takes 2 years or 3 years. But here, the protein, not only protein, but you can protein interaction, a protein other molecule interaction to form a complex. You can look at through this particular microscopy, cryo-microscopy, without preparing the crystals. So that's why they got the Nobel Prize. So many important. And let's compare this. This is the structure X-ray crystallography. You can see the resolution, nuclear magnetic resonance. You get picture like this, and you have to have many, conditions for that. Take a large amount of samples. Here you have to prepare the crystals, but if you do the cryo-microscopy, the sample is just frozen in native state, and you can see the resolution. So this is almost near atomic resolution can be obtained from for a big molecule, a large molecule, macromolecules like proteins or other polymers, and their interaction with other molecules, complexes. So you can get this very important discovery in science, in chemistry, rather in physics or biology also. This is, I want to show you a picture. Electron microscope resolution. This is resolution before 2013, doing the crystallography. But now we can see the atomic resolution by doing this cryo-electron microscopy, and at atomic visible or visualized proteins at atomic resolution. That's so important. You can see every part of the protein and try to find out the function. Now people are knowing, different functions of different region, because they got the structure in the atomic scale. And then if you bind this protein with other molecules or other proteins, then what will be the structure? You can find it. What is the binding region? You can find out. So many important. That's why the Nobel Prize going to the microscopic discoverer. And a lot of laboratories are doing this. And this is kind of, apparatus. I'm not going details of this. Cryo-EM, you have to have vitrified, solution, water maybe, and then you can sample very quickly to freeze the sample and then get the microscopy done. For example, in our lab, we do the phosphorescence. We do that, we take in the glycerol medium, 30%, 40%, and if you freeze it, you get the clear glass. And glass is very easy to do the spectroscopy. Similarly, here something we've done so that you can do the microscopy at the atomic scale. So important, without doing anything like crystallization. Crystallization takes place, sometimes 2 years, 3 years, very difficult. So cryo name stems from the low temperature, liquid nitrogen temperature, or maybe lower than that. This is the structure of Zika virus obtained from cryo microscopy. The structure of Zika virus solved in the cryo in just a few months using cryo-EM, and searching for possible sites that could be targeted by drugs to prevent the spread of the virus. So that's so important. If you know the structure in details, then you can find the right kind of drug and target the drugs to bind somewhere. So this is so important. And also, this pandemic period, I want to tell you that coronavirus has been also done under this particular microscopy, cryo-electron microscopy, and we get lots of detailed structure. And people are working on this. What kind of drug we use for this, so that you can destroy certain part of this virus. It cannot be, cannot attack our body or something. If you attack our body, you can destroy this virus. So you're searching for this drug. But if you search for the drug, you have to know the structure of the virus in details, in atomic scale, the various all the regions. Then you can try to find out what you should do. To kill the virus, or to prevent the spread of the virus. So that's kind of cryo microscopy is so important. Cryo-electron microscopy, along with bioinformatics, lots of paper and people are coming out these days. Results that will be, I think, fruitful within 6 months or a year. I hope that we'll be able to fight this coronavirus. And as I mentioned, studying the molecular mechanism, various life processes. Nanoparticles research, structure-based design, material characterization, everything can be done with this cryo-electron microscope. So important And single molecule spectroscopy, as I mentioned, these are some of the, calibration of molecular transistor, think of. And then I'm not going to details of this, detecting single molecule absorption, a very important area of research going on. And the single molecule imaging and spectroscopy, people get the idea doing the single molecule spectroscopy, and then they can apply in the imaging technique. This is completely different type of and very difficult kind of thing, because when you have to look at the single atom or single molecule. So that's not an easy thing, but people have done this. I'm not going to details of this, but the results are very important these days. A lot of people are doing this, but once you get the result, you can use this for various purposes. There's some surprises from single molecule. This is optically induced spectral shifts, something called Poisson kinetics observed. And molecule spontaneously jumps in frequency space due to nearby host dynamics. There are the works that has been done recently. For example, this dye laser, and so various kinds of work are done these days in the single molecule using the ultra fast lasers, and then you have to find this, that you have to have a right kind of camera and then right kind of software to find out all this. So physics people, chemistry people, computer people, all are doing simultaneously this research. So big research is going on this single molecule, single cell as well, as I mentioned. And optical imaging complex, what is the remove ensemble averaging? That's the one idea. And image detect nanometer scale interactions, and commercial sequence also used in the sequence of DNA or imaging, packed biosequencing, single photon courses, super resolution microscopes. So this single molecular, single cell spectroscopy has opened up a huge area of research where we can, detect or function of a particular molecule, and then probe particular area and get a particular right kind of drug, right kind of other ligands to find out certain solutions, certain diseases. For example, here sequence DNA or imaging, so done by this kind of spectroscopic techniques. I'm not going to details of this. There's another single molecule things. Again, this kind of emitted fluorescent room temperature. This is the impact. Chemistry, physics, biology, everywhere. Chemistry, spectral diffusion, intersystem crossing, molecular distortion, nano antennas, structures of materials, photocontrol, catalysis. Physics, you can call it nano environments, magnetic interactions, diffusion, structure, EM enhancements, quantum optics. Biology, you can talk about fluctuations, enzymatic states, mechanisms, cell biology, folding, protein folding, defolding, all this, membrane behavior, cellular structures beyond the diffraction limit. So these kind of things are, you can explore by doing single molecule spectroscopy. There's a lot of impact. Spectroscopy as well as microscopy has done a seminal contribution to our mankind, various problems. Then the final thing is, and to say about what is artificial intelligence and chemistry. A mix of mind and machine, a creative collaboration, design, make, analyze, test. So what is artificial intelligence? Probably the participants are familiar with what is artificial intelligence or machine learning or big data analytics, big data. And they are applied hugely in chemistry and biology, both sides. So this is, something cartoons, you can see that hypothesis, test, and end synthesis. I'm moving from chemistry synthesis, testing. Now, what is the idea of this machine learning or AI in chemistry? So this is chemical compound space, high throughput and quantum mechanical calculations. You can QM based databases. You have extensive and comprehensive QM data, analysis, chemical knowledge, and then you can get this insight into CCS, means chemical compound space. That means you have thousands of molecules and you can get the data using machine learning or AI, artificial intelligence. Say you have to find a particular drug, then we have the data of thousands of molecules, and you can think of which molecule is suitable. And that is a huge task, but AI and machine learning has done that, in a software have been developed or discovered. And we have found that this kind of molecules we can choose out of thousands of 10,000s of molecules, and we can work on that synthesis as well as their binding. And so in the drug area, this is a very, very important thing. It's a new artificial intelligence model to bridge biology and chemistry, because this is in silico medicine company, frontiers in pharmacology. As I mentioned, this artificial intelligence and this machine learning data analysis, this is the molecular encoder, differential expression encoder, gene expression changes. So you can everything you can, if you have the data, large set of data, then you can find out which data you have particular things to solve, particular problems to solve. And that's how artificial intelligence machine learning is helping chemists and biologists together, that they can work together. And these are the few things that in drug design and drug discovery. I am giving importance to this particular slide because now drug design and drug discovery is so important in this last year and this year. People are trying very hard. We are, our life is dependent on this people, the chemistry people, the microbiologists, the virologists who are trying to get the vaccines, but chemistry people along with the pharmacy people, they're trying to get the right kind of drug. And you see that, and artificial intelligence, identification of drug doses and drug delivery effectiveness, prediction of protein folding and protein-protein interaction, drug repurposing and quantitative structure, activity relationship modeling, prediction of bioactivity and identification of physicochemical properties, prediction of mode of action and admit analysis coordinates. And then there is peptide synthesis, small molecule design, de novo drug designing. You can see there is a classification between active and inactive ligands, monitoring the drug release. You can do that, which is active, which is not active, structure-based virtual screening. That is very important. If you can screening something, ligand-based virtual screening as well as structure-based virtual screening. Then biomarker discovery, preclinical and clinical development, identification of molecular pathways and polypharmacology, primary and secondary drug screening, and all these comes from this artificial intelligence, machine learning, deep learning. You can convert it to meta learning or generative adversarial network. You have the reinforcement learning. You have neural network. From there, you can simulate learning. You can support vector system machine. So people are using all kinds of things to get a right kind of drug. The main purpose is to get a right kind of drug. Several out of 1,000 molecules. So you have the property, you have the their functions, and they choose a particular drug. So it's so important in these days. So that is the importance of AI and machine learning. And these days, AI and machine learning has so much importance to find out, as I mentioned, right kind of molecule, maybe drug or maybe for imaging technique, maybe for other technique. So they are working very hard. Many chemists actually working on this. That's why the chemist argument is so important. And lastly, I want to say what is IUPAC induced top 10 emerging technologies in chemistry. And IUPAC, all of you know, IUPAC has, because this technologies project is to showcase the value of chemistry and to inform the general public as to how the chemical sciences contribute to the well-being of the society and the sustainability of planet Earth. And 2020, 21 has not been published yet. 2020, they say these are the things. One is aggregation-induced emission. We are familiar with emission of molecules, but sometimes if there is an aggregation, then there is a kind of emission, different kind of emission, sometimes called a white light emission or things like that. Those are so important area. AI applied to chemistry the last year. Then dual-ion batteries, as I mentioned, not only lithium batteries, but sodium lithium batteries or potassium lithium batteries are so important. Lithium is costly. People are trying very hard to get a sodium battery or potassium battery or using dual-ion, like sodium and lithium and potassium. So they are very effective for the car purpose, for the other purpose, even for bio purpose, that is, can be introduced in our body so that they can be, biocompatible. Then high pressure inorganic chemistry to synthesize many important chemicals. That comes under list of 10 within 10, the list of very important area of research. Then liquid gating technology, this is another thing that comes so important these days. apart from that, macro monomers for better plastic recycling. This is very important, because we are suffering from, this environmental pollution causing by plastics bottles. So people are trying very hard to get plastics which is biodegradable, but also people are trying to try macro monomers for better plastic recycling. The plastic as well as recycling process, and plastic made of materials which would be biodegradable. So that's a large area and very huge area of research people are going on, because otherwise, using the plastics will be a detrimental for our future, for the mankind. So you have to remove the plastics as far as possible. It's not possible always, so you have to find out the plastic which is biodegradable. Then microbiome and bioactive compounds. That's also, as I mentioned, bioactive compounds is very important. Imaging purpose, say we do the MRI scan. All of you are familiar with this, but MRI, you need some imaging materials or compounds and act as contrast or act as for imaging of various parts of our body. So that's another area of these bioactive compounds that we prepare. And the nanosensors, as I mentioned initially, nanomaterials can act as sensors. So people are trying to get nanosensors which are biocompatible, so that they can detect a particular area, for example, area of cancer, where the cancer is, where the tumor is, location of how the tumor is, or what is the area, or what is the particular part of body that has been affected. So use the nanosensors, a lot of people. And rapid diagnostic for testing, this is a very familiar word in this particular year, last year. And of course, RNA vaccines, because we are taking vaccines. Most of us are taking the Covishield or co-vaccines. These are old vaccines prepared by old method. But RNA vaccine is a new one, and that's why it come under these 10 important things. When we are unlucky to get this RNA vaccine, so Moderna is doing this, or Pfizer is doing this. Hopefully, it will come to India next year. People are trying to get this vaccine. A little bit costly. That's why we are not getting it yet. And one more important thing, RNA vaccine is somewhat unstable. You cannot store it at room temperature or even at freeze temperature. You have to have a 77 degree Kelvin initially. But nowadays, they have found a device which, you can keep in minus 20 degree or something, so that you can use a normal freeze. So that's how the research is going, whether you can have RNA. Otherwise, you cannot transport RNA vaccines. And transportation and, preservation is a very important problem. So we can solve that. We can keep the vaccines, because this is a very unstable thing. If you can keep that in minus 15, minus 20 degrees, then it will be available easily. And hopefully, we'll get that vaccine next year to protect ourselves and protect the all humankind, and purposes of, vaccines is so important. Okay, with this, I think I have told many things briefly. And finally, I want to say, all the participants, particularly the students or the new researchers, try to learn dreaming, because dreaming is one thing that if you dream, then it is the dreamers who will create the new science, new knowledge of tomorrow. If you don't know how to dream, then you cannot discover. You have to be imaginative. You have to be dreamer. You have to have intuition in yourself, self-confidence, all these things. But first and foremost things, you have to learn how to dream. And this is a picture. This is a picture of, I told lots of about single molecule spectroscopy or other spectroscopy. And this is a spectroscopy, means interaction of light with the molecules. But this is the interaction of light with Kanchenjunga. I am a fond of photo, and I took this picture when I went to Darjeeling. And if you are a early riser, then you get the red thing, Kanchenjunga peak, red, but you have to early rise before the sunrise. This is the color of the Kanchenjunga, red, completely red, the interaction of light with Kanchenjunga. And then if you get little bit, when the sun rises, as sun rises the color changes from red to yellow. And if you are a late riser, then you only see this white thing. If you get up at 7 o'clock or 6:30, you get this white thing, nothing else. But if you can get up at 5 o'clock, you can get this time resolved picture of Kanchenjunga. It is so beautiful in nature. Anyway, so if you look at the Kanchenjunga or other mountains, I should say, then you will be dreamer. You will be thinking something else. So with these few words, thank you all for your attention. Thank you.
thank you very much.